Good afternoon. My name is Lauren Wood Heickman, and I am a medical officer in the Division of Diabetes, Lipid Disorders, and Obesity at the Center for Drug Evaluation and Research at the U.S. Food and Drug Administration. The purpose of my talk today is to discuss considerations for surrogate endpoints and present what FDA considers to be validated clinical outcome measures in type 1 diabetes for the purpose of regulatory decision-making. Please note the views expressed in this presentation are mine and should not necessarily be interpreted as those of the FDA. I have no financial conflicts of interest to report. The FDA recognizes the need for more endpoints for new onset type 1 diabetes, which meet the standards for use in regulatory decision-making. I will first discuss the primary endpoints that have been used in diabetes trials to support approval or labeling claims for diabetes drugs. These include hemoglobin A1C, a validated surrogate endpoint, and severe hypoglycemia, a clinical endpoint. To recap on my FDA colleague Kristen Placino's discussion, surrogate endpoints are characterized by the level of clinical validation, and surrogate endpoints that have undergone adequate evaluation are called validated surrogate endpoints. These endpoints are accepted by the FDA as evidence of benefit and can be used in lieu of a clinical endpoint for traditional approval. Hemoglobin A1C is an example of a validated surrogate endpoint with a level of evidence that supports regulatory use. When qualifying a surrogate endpoint, the FDA considers strength and consistency of epidemiologic data supporting the relationship between the surrogate and the clinical outcome of interest. Generally speaking, there is greatest confidence in a surrogate when there are data from intervention trials showing that the treatment effects on a surrogate endpoint reliably predict treatment effects on a clinical outcome. This confidence is strengthened when this benefit is demonstrated in other drugs within the same class or drugs from distinct pharmacologic classes, regardless of the mechanism of action. Hemoglobin A1C is considered a validated surrogate endpoint as the Diabetes Control and Complications Trial, a prospective controlled clinical trial, definitively established the causal link that glucose control by lowering A1C can reduce the risk of disabling microvascular complications due to diabetes. Hence, hemoglobin A1C can stand in for the clinical outcome of microvascular complications for both type 1 and type 2 diabetes. Here, I list several strengths of the A1C assay as a surrogate endpoint. Usually, we use a surrogate when the real endpoint is relatively rare or is very delayed, making it hard to study. In this case, hemoglobin A1C prevents the need for a study designed to measure long-term complications, and the benefits of a drug can be established in smaller and shorter-term trials, bringing patients available drugs sooner. A favorable effect on hemoglobin A1C is considered to be highly likely to predict the clinical benefit of the drug, and therefore, a drug approved based on hemoglobin A1C does not require additional evidence of effectiveness for approval. In addition, A1C is a standardized biomarker, and adoption of standard methodologies set forth by the National Glycohemoglobin Standardization Program have allowed the assay to be precise to a degree that allows for use with regulatory decision-making. The agency recognizes there are potential limitations to any surrogate marker. For surrogates, the degree of uncertainty around the benefits of drugs approved on a surrogate endpoint are higher than for drugs approved on the basis of the directly measured long-term outcome. In addition, hemoglobin A1C does not capture glycemic variability. We also acknowledge there may be unique considerations when using hemoglobin A1C to demonstrate superiority as a surrogate endpoint for the type 1 diabetes new onset population. For example, a study designed with primary endpoint of hemoglobin A1C lowering may not be able to detect a difference between treatment groups because lowering A1C may be achievable in both treatment groups, given the frequency of follow-up in a trial, the standard of aggressive insulin management, and the physiology unique to the new onset period, like the honeymoon. I will now move on to a discussion on hypoglycemia. Severe hypoglycemia is characterized by altered mental and/or physical status requiring assistance and does not rely on a biomarker for diagnosis. Severe hypoglycemia is a clinical endpoint because it directly describes how a patient feels or functions. Other hypoglycemia is also mentioned here, with level 1 and level 2 hypoglycemia defined according to the American Diabetes Association definitions. Note that each definition contains a glycemic cutoff. If a specific blood glucose cutoff is used to define an endpoint, the definition of hypoglycemia moves away from a clinical outcome measure, how a patient feels, functions, or survives, and towards a surrogate biomarker-based measure. For example, not all patients with blood glucose less than 70 have symptoms. For surrogate measures, there needs to be consistent evidence linking a certain cutoff with a clinical outcome. That being said, there's building evidence to support level 2 hypoglycemia as a surrogate endpoint, defined as a blood glucose less than 54 milligrams per deciliter on a glucometer for patients with diabetes using insulin. In contrast, more work is needed to validate CGM-based hypoglycemia metrics for regulatory decision-making. Similar to using hemoglobin A1C as an endpoint in new onset trials, there may be limitations and challenges with using severe hypoglycemia as an endpoint. Studies in new or recent onset type 1 diabetes often enroll patients at a lower inherent risk of hypoglycemia. There may be challenges with trial design if the event rates are uncertain, and with low event rates for the outcome of interest, larger trials or longer duration trials might be needed. In addition to limitations for trials designed to detect severe hypoglycemia, there are also considerations relating to using non-severe hypoglycemia endpoints. Most importantly, these endpoints are not currently validated for regulatory decision-making. There is also variable performance among different glucometer devices. These devices also generally have poorer accuracy at the lower end of the glucose range, making it challenging to accurately capture hypoglycemic events. To summarize, the primary endpoints that have been used in diabetes trials to support approval or labeling claims for the diabetes drugs include hemoglobin A1C and severe hypoglycemia. Although FDA relies on hemoglobin A1C reduction and reduction in incidence of severe hypoglycemia to establish the clinical benefit of drugs in patients with diabetes, FDA recognizes reduction in hypoglycemia and in long-term risks are not the only clinical benefits important to patients with type 1 diabetes. In addition, although these endpoints have been used for approvals in the past, and hemoglobin A1C is the basis for essentially all glycemic approvals for drugs intended to improve glycemic control in both type 1 and type 2 diabetes, we recognize that there are some limitations to applying these endpoints in new onset type 1 diabetes trial designs. We note, however, that any outcome that can be shown to meaningfully impact how a patient feels or functions is considered a clinical benefit and can potentially serve as an endpoint in adequate and well-controlled clinical trials. We look forward to hearing comments regarding this important topic at the upcoming panel discussion. Thank you for your time and consideration.